All right. Hello, everyone. I am Aimee of AimeenRop.com. And as promised, I'm coming to you with the research in support of postnatal oogenesis, which basically means research in support of the notion or the scientific evidence behind the fact that we are making new eggs. We are not born with a finite pool of eggs. Now, that is not to say, and nowhere in my post did I say that that means women have eggs throughout their whole life like men have sperm. However, there is research showing that even in menopause, there still is around 1,000 or so eggs left in the ovaries. So we aren't really running out of eggs. This is all about the fact that we're not born with a finite pool that just continuously runs out with each cycle. That's what the post was about. I apologize if that triggered you in any way, shape, or form. My goal is always to educate, inspire, empower. And I do see a part of my path as being one that challenges the standard dogmas. I feel there is so much shame and disempowerment, especially in the space of fertility. And I am always looking at research and always trying to educate and empower my patients so that we start to question these truths, which, as you'll see, we'll dig into the original research that decided we have a finite pool. And it was done on mice and rhesus monkeys, just so everybody's aware. And we'll get into kind of using mice in research and why. And there is research not supporting postnatal oogenesis, but the research supporting it far outweighs the research not supporting it. And the idea, obviously, is that there will be a consensus at some point soon to really challenge this dogma that we are born with a finite pool of eggs. That comes from research in the early 1900s. Zuckerman was one of the lead researchers on this, and I actually have that paper that I'm going to go through with you guys. But let's get into it. Let's do it. Okay, so just so you know, and I did post it twice in stories, and I'll show it to you guys here too. I've created a Google Drive folder with all of the research used in this presentation. You can email my team. I'm going to create a pretty link for the folder. I think I'll make it be aimeerop.com/PNO, postnatal oogenesis. I haven't done that yet, but I will. But I have PDFs that I couldn't get all the PDFs because just not all the PDFs are available for download with the service that I use to access my research. But they are the full studies. The ones that I could get my hands on, I put in there, and the ones that I thought were important. I also put in there some of the ones that are showing against postnatal oogenesis, because that is what is called peer review, right? It's scrutiny. It's meant for scientists and researchers and medical professionals to come together and to agree or disagree in a professional manner. But so just so you know, this Google Drive folder, and I've actually been adding things to it as I'm doing more research. And I wanted to, I'll pull out the article too by Kaya at Fundamental Nourishment as well. And I'll do that while we're live here. It's easy enough for me to do because I subscribe to her Substack. And I've also subscribed to Jen Gunther's, I think that's her name, Substack as well. So I could read her piece, of course. Okay. And so again, I'm sorry if my post triggered any of you. My intention is to educate, empower, encourage bigger thinking in a system that is dated. And I would say in a system that is not in support of women's empowerment. And that's maybe a bigger discussion for another time. But if you guys have been following me for some time or have read any of my books or do follow any of the, the free advice that I give out there, which is a plethora of free advice. I've been sharing information on social media for at least 5, 6, I think 7 years. I can see my newborn in my hands, and he's going to be 8. So, almost 8 years that my YouTube channel, my Instagram, my Facebook, tons of free content. And I have many women that reach out to me that just consumed all my free stuff and had success with getting to their babies. We share that information constantly in social media as well. We constantly are screenshotting. We constantly are getting these women to come on and do stories of hope. You guys can go and dig into all of that. And that at this point is my social proof of my success, me and these women live on these videos, testimonials. We have gathered some data from my Yes, You Can Get Pregnant e-course and seeing the success rate in those women that come in, use the program, use the community, and about 65% of them actually have either 20 plus weeks. I don't count pregnancies until we're past 20 weeks, 20 plus weeks, healthy children in their belly or babies in their arms. And that research is actually on my site as well. And so, yeah, but again, I'm never, you guys are suffering enough and this journey is hard and I don't understand it to the level that you understand it. I myself have gone through loss. I understand that. I myself got pregnant naturally in my 40s. I understand that battle, the advanced maternal age battle. I understand that. And I have, you know, I've been on the journey, standing alongside in solidarity with literally at this point, thousands of women. And so I never want to add more stress to your plate. And I'm sorry if I did that. And I'm sorry if I was triggering. And I'm sorry if that's created doubt about my integrity as an individual, which was never my intention. And that's why I'm doing this, to show you where I came up with the data and why, so you can understand my thinking on the side, and also understand that this conversation about postnatal oogenesis is a debate that has been going on for 20 plus years. Some even say 70 years, since the 1950s. And it's a heated debate. It's super polarizing. I've been in medical research. I was a research scientist for a few years, undergrad and graduate programs, before I became an acupuncturist. And then even in acupuncture school, I worked with the research team writing grants and also doing research in hospice and on palliative care and pain management in hospice patients. But I've been in the lab, you know, studying rat brain cells. I did rat hippocampal cells, Alzheimer's research. I did that for 3 years. And I've been on the clinical side of things. I did stroke research when I was in college studying biology and chemistry. I worked at Kessler Institute in their neuroscience department. And then post college, I was in 2 different graduate programs. I did more lab research. And then in graduate school, again, I was basically in school until I was 30, something like that. Is that true? No, 28, because I'm 47 now. And in graduate school, again, for Chinese medicine, which is a 4-year Master's of Science degree program, I also did research. And then there is a doctoral program. I do not have my doctorate in traditional Chinese medicine. I chose to do other things than go back to school and finish that doctoral program, which is only 5 more, 5 semesters, about a year and a half of work. But I have been asked to teach at the doctoral program in my alma mater, and I have done that as well in the space of fertility, reproductive immunology, diet, nutrition, things of that nature. So let's get into it. These are my credentials. I kind of just went through them, but just so you know, because my integrity has been questioned, there has been defamation going on, and there is, you know, what I consider slandering of me as a human versus peer review, which should just say, we disagree, here's why, and give me the opportunity to also defend myself. So I have a bachelor's of biology with a minor in chemistry. That's from Rutgers University. Then during my time at Rutgers, I was a research associate at Kessler Institute. I worked under Dr. Bruce Diamond. Then I did graduate level work in microbiology at SUNY College and neuroscience at UCSD. My plan was to always go to medical school. The first time I took my MCATs, I did not do well. Standardized tests and Aimee don't usually work well. I've always been a really great academic student. I think I graduated college with like 3.4 GPA. I was a 4.0 in my graduate work. Across the board, every institution I was at. But I didn't finish either of these microbiology or neuroscience degrees, because at the time then I was planning to retake the MCAT, which I did, and then go into a PhD MD program at UCSD. That's how I wound up out there. And I was working in the lab of Dr. Arnold, or known as Arnie Miller, who was on, tenured staff at UCSD for, I don't know, 20, 30 years. Brilliant guy. We did research on Alzheimer's disease. And the Kessler Institute, I was doing stroke research. That was actually clinical research. So we were meeting with patients. We were looking at, it was women, postmenopausal women and stroke. Fascinating research about birth control pill use and not. Anyway, that was then. I was actually at Kessler when Christopher Reeves was rehabbing too, which was a cool time to be there. And when I was at UCSD, I was also a junior research scientist. Like I was legitimately on payroll at UCSD for about 3 years. And then when I decided to go into traditional Chinese medicine school, which was around the corner from the University of California, San Diego, I went to school in San Diego. I was out there 8 years or so. I stayed on as a research associate at UCSD. And then I also started helping Pacific College of Oriental Medicine get into research. So grant writing, research design, we were, like I said, we were doing work in hospice with patients, pain management, palliative medicine. Then I also have written 4 books over the last 11, 12 years. And anybody who's written books or has read any of my books knows that it comes with a good amount of research. And then also, I think there was question around my clinical experience. So I've been in clinical practice, and this doesn't count my graduate work, which in grad school at Pacific College of Oriental Medicine, it's a 4-year master's program. We're in the clinic from year 1. So I accumulated, I don't know, a couple thousand clinical hours before I graduated. But then once I graduated, I had 2 practices. I now have 3, but I had 2 practices. And one of my 3rd practice now is actually in a reproductive endocrinology office up here in Westport, Connecticut. I work side by side with reproductive endocrinologists and have done that for 2 years. And prior, I've had previous relationships with many REs. But clinical practice from 2004 to present. So I'm about to hit 20 years in clinical practice. And up until COVID, I was going to each clinic 2 days a week. And on average, I would see 15 patients a day, 60 patients a week. Plus I have, at this point, I have 4 associates that work under me in the clinics. And I also do case reviews with them on a weekly basis. We also host grand rounds where we're constantly looking at data, at how to support our patients. And because of my Western background experience and also the clinical experience I've been in, and I'm studying functional medicine as well at the School of Applied Functional Medicine. So we look at the whole picture. We're not just doing Chinese medicine. I am an herbalist as well. I study with an herbal mentor. I have done that. That's a graduate mentorship program that I've been in for 5 years at this point. In about 2016, I started online consulting. People started reaching out to me. They had read Yes, You Can Get Pregnant. So I started working with women. And currently that's my main job, is I do a lot of virtual work now. I only go to my clinics. I go to New York City once a month, and I go to Nyack once a month, and I go to Westport once a month. And everything else is run by my associates. I still do my case reviews with them weekly, and then we do grand rounds. And then my continuing education, functional medicine, nutrition, academic research. Again, I've taught at the doctoral level and working in relationships with multiple reproductive endocrinologists. Oh dear, I skipped all the way ahead. Sorry, that's really funny. Let's go back here. Okay, so one thing I wanted to get into is, why are mice used in research studies? Mice, because there was a lot of feedback of like, we're not mice, you can't share mice studies. The world of research always starts with mice. Mice fill a special and important role in medical research. Like humans, mice are mammals, and their bodies undergo many similar processes, such as aging. They have similar immune responses, infection responses, disease responses, and their hormones are similar. So that is why mice are used, and that is why some of the data presents with mice. It's not, it's almost like the starting point. Then once they see that, then they move to the human model. But some of this research is, it needs to be proven first in the animal model before moving to the human model, but mammalian animal models, because the consensus is that mammals are similar. Now, I agree. I never once said that we were going to be like naked mole rats. That was never what I said. I shared research showing how naked mole rats have fertility forever, because that research is how we're learning comparatively what are humans doing versus naked mole rats. Nowhere did I insinuate that we are naked mole rats, just to clarify that. And I don't want to go on the defense. I don't need to go on the defense. I feel very solid in the human I am and in the data that I present to my community. But it's not fun to be slandered. Okay, please also note, so I'm going to show you this study next, that the original studies dating back to the early 1900s, again, that's why I put in that study. I was trying to show that even in the 1900s, this debate was going on. People were saying, do we really have an infinite pool of eggs? Do we not have an infinite pool of eggs? And you'll see too in the data, they just use mathematical models to determine whether or not we were running out of eggs. So where was women run out of eggs on mammals, and the main research was on mice and rhesus monkeys. And I also wanted to point out that the research that you guys all love taking NMN now, True Niagen, NAD, you name it, Elysium, anything for anti-aging, you know, Dr. David Sinclair wrote his book, How We Age and Why We Don't Have to. And he has this NAD studies in there showing that mice come out of menopause and make healthy eggs went on NAD, and now everybody's prescribing that. But that's mice studies, just so you know. So a lot of studies on supplements are also done on mice. Not all of it, you know, like CoQ10 and melatonin, those studies are actually on humans and women undergoing fertility treatments. So, but just, I just want to point that out, that this is a normal thing. This isn't, you know, me cherry picking data to show that mice are creating, have postnatal oogenesis going on and not showing the human data. There's a lot of human data, and I'm going to go through it. Okay. So, and lastly, before I get into showing the research papers, I want to just go over what peer review is and what peer review means, because what has just happened on social media is not peer review. Peer review involves subjecting the author, which I was the author of that post, to scrutiny. That's fine. I disagree with this. Show me more research. Can I have you on my page? Let's talk about this. This is an interesting topic. It keeps coming up. Check for validity, evaluate suitability for publication. A peer review helps the publisher decide whether a work should be accepted or not. Now, I am not a medical researcher. There is freedom of speech. I can put my posts up, right? Unfortunately, we aren't that managed. And that, listen, there's plenty of posts that I see that I'm like, oh gosh, there was one recently from a peer who is a doctor that I disagreed with. And I went into her comments and I said, I highly respect you. I admire you. We cross refer all the time. She's a reproductive endocrinologist. She refers to me. I refer to her. I disagree with this. Based on the current research, I disagree with this. She sent me a DM. She said, thank you so much for being so professional, Aimee. I really appreciate that. We should chat this through. I would love to see the research that you saw. That's peer review. That's supportive. That's kind. Okay, so let me just see. I think I skipped ahead again. Let me just see. No, I didn't. Okay. Just want to see something. So I moved these around, but I think, yeah. Okay. What I'm going to do is I'm going to close this presentation down, and I want to go into, oh, I see what I did. They're over here. This is the first thing. So there's a lot of research. You can see every single thing open up here on my desktop is research. These are all, I use a website called Deep Dive, where I get access to all my medical journals. So you can see, and I get to go in and highlight. It's really a fun, it's $49 a month. It's very reasonable for someone who likes to read research, because you can see one PDF all on its own can be $40. So I also have a lot of medical doctor friends that will get me research papers if I can't get my hands on it. So that's easy for me. I feel grateful. Originally, I did record this yesterday, and I started with the oldest science, and then I decided maybe I should start with the newest reviews, and then we work backwards. But I do want to first show you, this is the original paper by Zuckerman, published in 1951. Okay, and I actually have it right here. We'll go into it, but I want to show you all his other paperwork. So I just searched his name, Zuckerman. right here in NLM. You guys can all do the same. This is public access. The only thing you can't get is you might not be able to get the full research article publication date. Let me just do this. Results is fine. So we'll go back. Why is it? I don't know why this is not actually showing up now. Either we're going to go back one. Okay. So we can start. Oh, that's what we do. We just click on his name and then all his papers will come up. But he's been quoted. This is how many times he's been cited. So he does not have 725 research papers, but you'll see every single paper up here cites this original research. So the relation of age to number of oocytes, that's the one I have open right now that we'll go through. The number of oocytes in ovarian fragments after compensatory hypertrophy, the number. So he did rat studies. Okay. Rats, adult rat, litter size, that's rats. So I just wanted to show this to you guys just so you understand the original research that has come up with the dogma that we run out of eggs every single cycle comes from the 1950s. This gentleman, Zuckerman, and his original studies were done on rats. Okay. The total number of oocytes in the ovaries of rats decreased with age. The relation between two variates confirming in the regression of oocyte numbers on age is defined by. So, and there was him and this other guy, Array, that both did some studies. And so, and basically what they did was they used the nucleus as the marker. So you can like stain and with fluorescence, or they were probably using not fluorescence back then, but some kind of assay to follow and track the number of eggs in the ovaries as the mouse aged. The other thing about mice is that they age faster than we do. So, you know, their days are kind of like our years. And so we can really see how age impacts them and quickly. So on our own data, it was derived from counts of both ovaries, 47 normal rats. 36 rats belong to 9 litters, ranging from 60 to 262 days. No rats under 35 days were studied. All estimates of small oocytes have been corrected to allow for diameter. As we have failed to find evidence that the total number of oocytes varies cyclically with the estrous cycle, no adjustment was made to allow for the fact that the animals were not killed in the same phase of the estrous cycle. I'm not sure. So I think they had to kill the animals and then they counted the eggs. That's what I'm thinking they did. We do it differently now. I mean, these mice unfortunately do die in droves, but we do it differently now where they'll do these assays and you can actually target specific cells. And you'll see that in the newer research. But so here's their studies of like, we'll just look at what this is, the interrelation of oocyte numbers and age in the rats. And so number of oocytes starts around 20,000 and declines to 1,000 was the cutoff. So not even, above 1,000, under 2,000 age in days. And I don't know the exact translation, but you guys could Google it of like, what's the comparison of a 100-day-old rat to, a human. It'll come up. The number of oocytes in the rat age 63 days was 4,637, and the rat aged 262 days was 2,364. The figures appear to be very different, but if they're standardized by means. So again, this is all rat animal studies. And even if we were to look at like ones that don't say rats, I think we could see that they did rat studies. So again, 1952. Yeah, so female rats again. So just so you know, the original research, I believe, was only done on rats. I'm not even sure it was extended to the human model. You guys are welcome to dive deeper into that. So that's one of the oldest studies. This is one of the newest studies. This came out March 2022. There's another one that is later than that. But what I love about this paper is, and I put it in your Google Drive folder, is this a summary of O.S.C. research for and against. So I actually made this smaller just so I could make a screenshot for my slideshow. So this sorting table, this is published studies that have used, so there's these antibodies that they can now target on the cells of both human and animal mammalian models to help them sort and isolate these oogonio stem cells. So green shadowed boxes show successful oogonio stem cell isolation. Yellow shaded boxes, successful oogonio stem cell isolation with questions about antibody specificity. Red shaded boxes, unsuccessful OSC isolation. So basically what this means is this is all the current research that was using this new technology to isolate these cells. This is all the stuff that's out there all the way back from 2009 up till now. And this is one that had a hard time identifying, and these are the 4 that didn't identify them. 1 was on humans, the other were, sorry, 2 were on humans, the other 2 were on mice. 1 did both, mouse and human. But you'll see in here, we have 1, 2, 3, 4, 5, 6, 7, 8, 9 studies that identified oogonio stem cells in humans. And then what's nice too is, I mean, you're going to have to like cut and paste this, but it is a Google Doc, so you should be able to just go like this. Sorry, let me do it. You would go like this. Oh my God, my mouse. Copy. You would go over here then, NLM, and you would find this paper. Stem cell support of oogenesis in the human. Okay, and this was published in 2009. And then here's the abstract. And then if you have, deep dive is the one I use, like I said, $49 a month, and you could go and you look, and I think I already have this one open, so I don't need to open it again. But either way, this is how I do my work too, is I will go and I will read these papers. And so the nice thing about review papers is that they review all the data out there. It is obviously subject to peer review, and it is obviously then published by a medical journal, which, I mean, to get published is not easy. And so they really fact check every single thing. So you guys have this now, it's in your Google Drive folder. It's right here, a summary of OSC research. But you can see just by the looks of this now, and I'm not saying that this is my be all end all, but just by the looks of this, I didn't even count. I only counted up the human studies. There are a lot of studies showing the presence of these oogonio stem cells, lots of them. And there's 4 that show that they're not. Now that still makes us question. I'm not on the side of, it's funny, in the field of reproductive biology, they actually, because this is such a polarized debate, they will say you're either an ovarian optimist or an ovarian pessimist. And so I am and always will be an ovarian optimist. And I was before I even saw that label. I just think it's kind of funny. Now, me and all my doctor friends are like, we're ovarian optimists, and I am. And there's a lot of research going on in stem cells and PRPs and all these things that I won't get into. But this was all the beginning of that research, and it's fascinating. And clinics outside the US are doing stem cells constantly, for POI. I just had a patient do it in Spain, about $4,000 US dollars. And the doctors believe she'll have a 40% chance that her POI is going to be restored. That's a significant chance. And so anyway, and I also want to go on and show you. So this is the ability to isolate these oogonio stem cells. Okay. Then they also go down and break down. These are published strategies other than DDX for antibody sorting that have been successfully used to isolate mammalian oogonio stem cells for analysis. And these are all the different ways they did it. And again, showing you everything here. And these were all studies that were able to do it. They didn't show you any studies that didn't. And then this one, contemporary studies since 2004, supporting or refuting the existence of oogoneal stem cells and or postnatal oogenesis in mammals. Green shadowed boxes support that L.Cs and or postnatal oogenesis. Red shaded boxes refute. Okay, so again, you can see it is heated. Again, we have every single one of these is in mice, so just keep that in mind too. It's not like these guys are studying it in humans and these guys aren't. This is the whole idea with research, academic research, is that you publish, like here's an example. Like if we go actually to this paper, workflow optimization, they down here, materials and methods, you have to publish exactly what you did so that some other lab can go and recreate the exact same thing. And then we get to see, that's peer review. We get to see, is that the result? Is that actually happening now in my lab over in North Carolina versus your lab in Indiana, right? Different scientists, different, but same mice, relatively speaking. And then also in the human studies. So the one study, and now I'm jumping around, the one study that was pointed out in this, that shows no oogoneal stem cells, which came out in March 2020, which is also pointed out here. That was against it. You'll find it. It's on here. I saw it, Wagner. Here it is, the Wagner one. So it's clearly on there. The one thing about this study that is, I mean, I think there's a couple things, and they go through it actually in their study. They point out all the potential reasons why they didn't find it, because this team actually found them once before. This Wagner, and I think it's Yoshishira. Let me just see. Was it Wagner et al? No, I could just search the doc, couldn't I? Let me just see, is it here? I know I saw it earlier. Or I have these guys. Anyway, we'll look it up and I'll show you how I would do that. But the argument against, and they actually point it out in here, they say all the reasons why. So although it is well understood that ovaries cease to function when women enter menopause at age 50 on average, there are reports suggesting that pre and postmenopausal adult ovaries contain stem cells. So, and then they cite all the stuff that's in support. They're enigmatic and have been debated, of course. These OSCs have been isolated from human ovarian tissue. So they're saying here in this study, yes, this has been isolated, and they're using this antibody here. And then they're also doing RNA helicase, which are these are just basically assays. That's how I would kind of keep them in your head, of like how to identify and target these cells. It all gets to be like very mumbo jumbo-y if you don't really sit with it all, and all these acronyms get overwhelming. Germline markers. So basically, but so these guys mapped stem cells, but what they did, it was 21 patients, which is a pretty small study. And this is what's interesting. They took 21 cesarean sections. Five were from tissue from cesarean sections, and 16 were from patients that were undergoing gender. I want to just say it right. I don't want to screw it up. It's right here. Where is it? Gender reassignment patients. So people that were using, basically they were using androgen therapy to go from a woman to a man. And so that's whose ovarian tissue is mainly used in this group. And these people were on androgen therapy prior to surgery for an average of 46 months, upwards 14 to 89 months. I think that poses a challenge with their data personally, and other people have pointed that out as well. And then also they're arguing that they didn't have a large enough data set. And they actually say all this in here, which, again, peer review, being very professional. These are the areas where we could have done better. This is what could have been why we didn't find what we found. And then it's always like, and then this is an area of extra research. But one of the studies was showing, I think it was this one, that on average, you need 90,000 cells to get 5 OSCs. So I want to start here, and I know we're already, I think I'm an hour into this already, my gosh. But I want to start here talking about, I just want to read this to you. A central underpinning of reproductive biology is held that oocyte generation in ovaries of female mammals is restricted to the embryonic period, meaning while we're in utero, right, as an embryo. This thinking deviates markedly from spermatogenesis in males throughout adult life, which involves meiotic differentiation, male germline, and germline stem cells in the testes. So men have sperm line stem cells, so spermatogonial stem cells, okay, SSCs, different than OSCs, oogonial, coming from the oocyte, which is the other word for egg in our ovaries. However, the longstanding paradigm of non-renewing oocyte pool was challenged by a study with mice in 2004, which reported the existence of a female germline, OSCs, right, and the continuation of oocyte production in adult mouse ovaries. While this study sparked significant debate, and then, so typically that's what I'll do, then I'll go to 4 and I'll go to 5 and I'll read these citations and I just kind of gather my information. More than 80 corroborating, so corroborating means different labs did the exact same thing and found the same thing, right? So corroborating studies now support, so 80 corroborating studies now support the existence of oogonio stem cells and or active oogenesis in adulthood across species, including humans. And then they have the summary tables, which I've showed you in here. That's the summary tables, okay? So these are their summary tables information too. This discovery of OSCs, which brings the biology of male and female gametogenesis in mammals more closely in line with one another, and with that of non-vertebrates, because non-vertebrates also have postnatal oogenesis, not topic for conversation right now, has significant ramifications for the development of in vitro models to investigate human oogenesis, as well as new technologies to combat ovarian failure and female infertility caused by aging or insults. And then you would go 17 through 19. So, and then this is a major breakthrough in the study of OSCs came in 2009 with the first report that the cells could be retrieved as a distinct population using dead box helicase. So this is basically, they sorted the cells. They were able to identify like, oh, there's this antibody on these cells, these OSC cells in mice. And so now they can sort for that. It's really cool. They can tag it, it's fluorescence and it glows. And so then they can actually kind of follow. And I think in here there is a picture. Let me just see. Yeah, where they cell sort OSC meiosis oocyte. So single RNA sequence analysis of human ovarian cortex. So they took stuff from the ovary cortex, reveals transcriptional profiles consistent with the presence of oogonio stem cells and active meiotic division. These findings reinforce and extend a growing body of work collectively demonstrating the existence and functional properties of OSCs, as well as the differentiation of these stems cells throughout meiosis to form new oocytes. Okay, so they go into, okay, now we can identify them. We can actually sort and we can identify. And that's what this guy's saying, is that they did this, this DDX4, where is it? They did the same mapping and yet couldn't find it. But then they go in to say why. Here it is, DDX4, data analysis included. So then they found 6 main cell populations. And I just want to, anyway, they go through it here. So this is one not in support, but again, there's 80 or so studies in support of this. Okay, so human OSCs express a profile of gene characteristic of primitive germ cells, and these cells can differentiate through meiosis into oocyte-like cells in vitro and into oocytes that are enclosed within newly formed follicles after transplantation into human ovarian cortical tissue. So they're doing a lot of these transplantation exercises, and they've isolated fluorescence activated. Okay, so, and then this is the study I think they're talking about. Let me just see, I'm on page 3. I just want to double check. 25 is the Wagner study. So now they bring up the Wagner study right here, this same one, that is saying we didn't find anything. And so we can go back to that. and we're going to read about it. So it says, discordant with this large body of work, a recent study concluded single-cell RNA sequence analysis that human OSCs did not exist. The authors identified only 6 clusters versus 6 cell types in adult human ovarian cortical tissue biopsies. So everything, the non-oocyte germ lineage were apparently missing from their analysis and was subsequently put forth as evidence of OSCs being absent. However, the expression and clustering were performed with an early version of Cell Ranger software, which is widely known limitations. So this is them. This is again peer review pointing out these are the things that could be wrong. Another issue that can affect the resolution was, let's see, all of these highly relevant because OSCs, like other stem cell types, are very rare. So basically they said maybe they didn't get enough, maybe they used the wrong software, because why did 80 studies actually show presence? And now this study, one study in how many of them? So there's the most recent, so 4 studies out of all these that showed the presence didn't. Why is that different? And so that's the stuff. So I mean, that's where, sure, I can see people on the other side of the debate saying, but there is data out there saying we can't find them. But yet there's a lot of data saying we are finding them. And so what we need is people to continue to repeat the studies. But at this point, and I'm a big follower and fan of this man. He is one of the leading researchers in this field. He's Northeastern University College of Science, Jonathan Tilley. He's a professor in biology, and he has, so many publications. But his main goal in life is to study female fertility. Currently, we have major research efforts directed at defining the role of biomechanics in germline stem cell differentiation, uncovering the developmental switches that govern oocyte formation in fetal versus postnatal life, mapping the mechanisms. So, I mean, this guy, and you can email him. I'm actually considering asking him to come on and do a live with me, and we talk about this. I haven't reached out to him yet, but that was recommended to me, and I think I will do that. I'm sure he'd be open to it. And he was just in the news in 2022. I actually posted this in my story the other day. You can go on here. I can put this into NIH grant, help scientists revolutionize fertility research for over 20 years. So this is 2017. Fertility research brings death of dogma, birth of hope. This is the dogma we're questioning. Do we run out of this infinite pool? Do we have a finite pool, or do we have postnatal oogenesis going on? And so you can go and look at his stuff. I mean, he's legit, these are all his publications. He's on faculty at Northeastern University, and let's see, my research funded by NIH since 1994. I'm an inventor of 10 issued patents, have more than 20 other patent applications. He's been in Time magazine. I mean, anyway, interesting guy. And you'll see he's on a lot of these research papers, although they're pointing out all the different labs. So you'll only see him, I think you'll see him mentioned Tilley here, Tilley here, Tilley here. So he's done 3 of them, but then all these other scientists have repeated his work and have found these oogonio stem cells. Again, hence my determination from the data that we are not running out of eggs with every cycle. And I'm not the one making this up. So they continue to go on and talk about the Wagner study and what could have gone wrong, if you say, but given the past debate over whether mammalian OSCs exist, and with our labs being directly involved in studies for nearly 2 decades, we felt it was important to experimentally assess the conclusions reached by Wagner. Again, this is healthy peer review. We are scrutinizing in an effort to reconcile this recent report with the opposite conclusions reached in over 60 other published studies that have isolated and characterized oogonio stem cells. In parallel, we evaluated the possibility that the technology could provide further evidence. And so So such an outcome, which has not been demonstrated in humans, would be consistent with recent genetic. So then now they're trying to basically replicate this studies, this guy who she quoted, and see if they came up with something different. Okay. So, and here's their, and they're doing the same thing, general cell types and clusters reported by Wagner. They're again quoting the study here that she quoted here. Notably, the 1,260 cells analyzed by Wagner was 14.4% of the minimal cell input number needed for detection. So they came up with a number, I had read it somewhere, something like 90,000, that we need 90,000 cells to detect 5 OSCs at 95% confidence. So they think they didn't have a big enough sample size. And then using our optimized analytical pipeline, 10 cells were identified. Let's see. So workflow adjustments to optimize. So basically what they did was try to optimize, this Wagner study to see if they could get different results, right? Analysis of human unsorted ovarian cortical cells for evidence of non-oocyte germ cells. So we moved on to the analysis. So they're continuing to analyze and redo this study. And then they found additional genes that were expressing germ cells, STAG, SMC3. So, and then this is just a, chart of all that. They found other things. So you guys can read through this, but they're finding all these different expressions of either antibodies or oocyte markers, positive expression of the genes indicated a dark shade purple. So in the Wagner study, the authors also reported DDX4 antibodies based on that's fluorescent symmetry, led to the isolation of PVCs, which are, I think, perivascular cells and not OSCs. With this information in mind, PVCs are known to express anti-fluorescent biomolecules. We therefore sorted dispersed ovarian cortical tissue using adult bovine ovarian cortical tissue for validation. So they're just comparing. And then a distinct population of anti-fluorescent events was similar detected in dispersed ovarian cortical tissue of reproductive age women. So that's the other thing too of, the ones going under their gender replacement also were reproductive age females, not to be discriminatory by any means, but just meaning biologically at this point in time. And so germ cells and PVC segregate into distinct clusters. So this is what they find. I know this is a lot. So here's our discussion. While this RNA sequence is a useful tool to gain insights into cell lineage heterogeneity within a sample, a major caveat of this approach is that its failure to detect gene expression based on evidence of a given cell type after clustering analysis does not, by default, equate to that cell being absent in the sample analyzed. This is especially apropos in attempts to identify either very rare cells, so these challenges were further complicated by the fact that they restricted their efforts to find evidence. Anyway, you can go on and read this if you want, but basically, thus, this RNA sequencing workflow design and any conclusions drawn based on the expression of a single gene, being detected or not in a cell of interest, lacks scientific rigor and confidence. However, optimization of this workflow pipeline using Cell Ranger 3, so better data reader, software, which was available to them, but was not used for their unsorted cell analysis, allowed us to identify rare cells in the adult human unsorted ovarian cortical cell data set with gene expression profile that closely aligns with the primitive germ cells, such as embryonic PGCs and adult ovary-derived OSCs. So further analysis shows that 2 of these non-oocyte germ cells express multiple markers of meiosis, commitment, and progression, meaning that they're committed and progressing to oocytes. So becoming X. These observations, which offer evidence for ongoing de novo, which means original, postnatal oogenesis in adult human ovaries under normal physiological conditions, are consistent with prior studies with mice, have demonstrated their resident germ cells routinely undergo meiosis in adult ovaries. Interpretation of caution must still be exercised here, however, since gene expression profiling does not offer definitive evidence of the existence or not of human OSCs or of active meiotic entry. In this regard, all scientific studies of isolated human OSCs published to date have characterized the cells following isolation by gene expression profiling, along with various downstream functional tests of meiotic cell division capability and or oocyte forming potential. Like other gene expression based studies, so what they're saying is the RNA sequencing isn't kind of the best interpretation. So that's what they're saying. Like, caution must still be exercised here. There's other methods of isolating these genes. We're not so sure that this is the best method. But functional verification of suspected lineage identity is another caveat of the use of this type of big data technology. And I think conclusion, since the initial report on OSCs almost 2 decades ago, over 80 primary research studies have been published supporting the existence of OSCs and or postnatal oogenesis in mammals. More than 30 of these have sorted OSCs from ovaries with polyclonal or monoclonal antibodies on the DDX4. That's the most popular thing. In the same time frame, only 10 primary research papers have been published disputing the existence of OSCs. So 80 have shown it, 10 are disputing it. Now, absence of evidence does not mean evidence of absence, right? So that's that's a common statement made in research. And so, yes, there are still 10 research studies that have disproven, but there's 80 that have proven. And so only 4 of these studies claim that DDX4 was failed to isolate OSCs. With respect to human OSCs, at least 6 different groups have established and independently corroborated the extracellular. So there's more science on the side of this happening than there is on the side of it not happening. But again, and you guys can actually draw from it your own conclusions. That's why there is a big debate here. And I have drawn my own conclusions from this because I have looked at all of these research papers. In consideration of this, along with experimental evidence presented herein, a more reasonable conclusion from the Wagner et al. Study is that RNA sequencing workflow used by the authors was not designed appropriately to identify candidate OSCs, or in fact, any stem cell type in their sample. When the analytical workflow was optimized and applied to all cells, their sample equally, we uncovered evidence in their data set of both OSCs and primitive germ cells. So they basically disprove this study that this Jen Guntner's piece, where she likes to put me down here, that she uses to, so this study on naked mole rats, again, is a comparison study. No, anyway. And then she just goes into naked mole rats. So some initial data emerged. So she does point that out, which is great. But she only goes up to 2004. She doesn't present studies all the way up to 2023 because there are studies up to 2023. And then she says it's inevitable, which I think that's a very bold statement as well, because the data is not suggesting that, or at least it's questioning it. So at the minimum should be a sentence that says more data to be collected to further understand the biology of the ovary and the oocyte and whether or not and so again, this is the Wagner study she's quoting, and then she puts in the naked mole rat study that we all used. This one, I'm not sure where it comes from. This is the same researchers. So she's using the same study, just published in a different area. Anyway. And again, she shared, I think, these 4 studies that I've also pointed out that did not find. One was in monkey, 2 were in human. So I'm going to stop here for now, and there is a few more studies I'd like to get into, but in the meantime, you can do that too. Some of you who are really interested in this, and I really appreciate that interest, have come to me and said, direct me to the ones that you think are the most important to read. This is another one that I think is great in aging. This one, 2022 Journal of Ovarian Research. Because populations around the world are aging fast, the issue of aging has become a growing concern. And they go into, again, fast seeds and ovarian aging. And they talk about all the different studies that are out there and the evidence. Kind of show you here. And there's a lot of talk in this article too about the ovarian niche, like the environment that allows for these stem cells to propagate. And look at this: chronic inflammation and oxidative stress impact these the stem cell senescence, so making them die faster. And that's also where people argue like you can't improve egg quality. Well, there's actually real clear data that if we reduce inflammation and reduce oxidative stress, we are improving quality. Again, up for debate, and I'd gladly start doing these on a monthly basis if this is of interest to people, because I look at the research all the time. If this is of interest to you, just in the comments below, go for it. And I'll come back and do more. I've now been at this an hour, and I'll do another hour on other data. But I think this research paper is actually a really interesting one, and it talks about oxidative stress, injury, aging, which I think a lot of you would find interesting. This is a very, sorry, it's this one, parthenogenic embryo-like structures in the human ovarian cell surface. Let me just Control-A, Control-C, Control-V. Here it is. I'm having a hard time for some reason